Welcome everyone who's joined today for Infex's second webinar on the broader pipeline and growth strategy. So I'm Louis Ash-Jepson, I'm a Senior Associate at Albrook PR. We are media advisors to Infex Therapeutics, and we're hosting today's webinar on their behalf. Just a little bit of housekeeping before we get started, and I hand over to the team. There's going to be a presentation first, and whilst that presentation is ongoing, you can submit any questions you have to the Q&A box at the bottom, and the team will get to those questions at the end. So without further ado, I'll hand over to Dr. Peter Jackson, CEO of Infex Therapeutics.
Thanks, Louis. Hello, everyone. I'm Pete Jackson. I'm the founder and chief exec of Infex Therapeutics. And I'd also like to welcome my colleagues, Professor Colm Leonard, who's our chief clinical officer, and Dr. Vicky Savage, our chief scientific officer. What we're going to do is just take you through a bit of background and the investment opportunity for the company. Vicky and Colm will then take you through the more presentations on the key three programs that we have in our pipeline, and then we'll finish off saying a bit more about the investor opportunity, and then take questions, as Louis already indicated. So just the introduction to the investment opportunity and to Infex in headlines. We're seeking a minimum of 10 million of new equity to support the next 3 years of development of the company and to deliver the key programs, and as well as commercial milestones. So Infex is dedicated to the development and licensing of novel anti-infective drugs, but focused on high-value opportunities meeting critical unmet patient need, and really importantly, to reduce the healthcare burden on insurers' healthcare systems. We have an exciting and diverse pipeline of novel therapeutics, mainly focused at treating WHO critical priority bacterial pathogens, as well as viruses of pandemic potential. And we have near-term developmental milestones on each of those programs. The three programs we'll focus on are, first of all, Respex, which is a first-in-class antivirulence antibody with blockbuster potential, so potential revenues in excess of $1 billion, which is currently in phase 2 clinical trials treating a disease called non-cystic fibrosis bronchiectasis, or NCFB. And we have on that program a license option agreement already in place with major pharma company Shionogi from Japan. The second program, Metex, is a drug targeting critical priority drug-resistant infections, so superbugs. And this is a resistance bypass drug. It switches off the resistance mechanism in an important class of superbugs. And in this one, we have the first regional license deal, the term sheet in negotiation at the moment. The third program we'd like to tell you about is Covex, which is a pan-coronavirus drug, but is a new target that is available to treat MERS, SARS, and all COVID-19 variants, and also multiple coronaviruses in animals with pandemic potential. We've received significant non-dilutive funding for this, and on this one again, a first license agreement for long COVID applications is currently in negotiation at term sheet. So overall, we have an experienced management team with a proven track record, a well-defined commercial strategy focused on executing valuable deals and pharma partners. And I should say this is the third biotech company that I've been working on in partnership with John Moulton. John is our largest investor, major investor, also chairs the company, and has got a great track record in commercializing life science opportunities. Just to say a little more about the Infex pipeline. So the three main programs we'll talk about in more detail. So Respex, antivirulence antibody, is currently recruiting in phase 2A, and you'll see there that we've received significant grant funding already for this program from ICON, the UK Infection Innovation Consortium. And as I mentioned earlier, we have a partnership agreement in place with Shionogi for with a license option for further development and commercialization. Metex is a small molecule oral treatment for these critical NBL infections, and Vicky will tell you much more about those. We've completed the preclinical development on that, so the program's coming up to be ready to initiate phase 1 clinical trials. This program was originally in-licensed the know-how for this from Swedish company Medivir. As I say, we have a license option agreement in place with another partner at the moment. The COVEX is an in-house program. This is a small molecule oral pan-coronavirus agent, and focused on, as I mentioned, pandemic preparedness and also long COVID. We've received grant funding here from Innovate UK, the UK business investment entity, as well as ICON. We've also have a contract with the US National Institute of Allergy and Infectious Disease, NIAID, to support this in preclinical development. And we have agreement from the UK Agile COVID-19 clinical development program that COVEX would be acceptable into that program. We also have a pipeline of other really interesting assets in early development. We won't be talking in detail about those at the moment, but you'll see there that we have relationships with Microbiotics in the US, Redex in the UK, and grant funding from Carbex, which is the US accelerator that specifically funds research into new treatments for these superbugs. And then finally, at the bottom, we have multiple programs currently in development with university partners, and those have supported by potential funding from ICON, but also from the new PACE AMR accelerator that's been launched in the UK. So now just to say something about the potential for these projects in the markets. So Respex is in the $45 billion chronic respiratory disease market. And within that, NCFB is currently around $3.5 billion and growing at 6.5% CAGR. The projected peak sales for Respex in the NCFB market is 1.1 billion, and sales would be up to 2038, and that would deliver cumulatively $1 billion potential revenue for Infex from licensing by 2040. And this is without potential further expansion into the CF, COPD, and acute infection markets. So acute infections such as ventilator-acquired pneumonia, hospital-acquired pneumonia, HAPVAP. Metex is a targeting superbugs, so antimicrobial resistance. The current market is 4.6 billion, very strongly growing due to the rapidly rising spread of resistant organisms from places like India, China into Western nations. We have an initial regional license term sheet at the moment, potential yearly sales in that market of $38 million, and the potential overall revenue from that license deal of up to 80 million in total by 2040. Now, there's been some really significant developments in how new antibiotics and AMR drugs like Metex might get paid for in the major markets. These are so-called pull incentive schemes. And at the moment, there are pull incentives in play in negotiation in the US, in the EU. And actually yesterday, the UK announced a permanent pull incentive scheme in the UK market. These would provide up to $400 million per annum for new AMR drugs for up to 10 years. So that's $4 billion in total. And a license income for Infex into those new pull incentives could deliver total income for Infex of up to $600 million over that 10-year period. And finally, on COVAX, everybody's fully aware of COVID-19 and the actually therapeutic drugs for that, so not including vaccines. The market peaked at $11 billion in 2022. So at the height of a pandemic, there's a huge opportunity here. Long COVID has been estimated to have an impact of $170 billion on the US economy in 2022. So effective treatments for long COVID are also a huge market. And to explore that, we have a first license agreement, again, at term sheet with a partner to look at long COVID therapeutics using COVAX in combination with other drugs. And the potential upfront milestone and royalty revenues from that deal would be in excess of $100 million. And then, obviously, the potential for pandemic preparedness is large. Because our drug covers all coronaviruses, there's potential for use and stockpiling in against further outbreaks of MERS, SARS, and COVID variants. So the potential for multi-billion revenues from the next pandemic as well. So I'd now like to hand over to Vicky and Colm, who will take you through the main 3 programs and give you more information on those.
Thanks, Pete. Yes, so introducing our first program, which we'll be presenting to you today, which is Resvex. So this is a monoclonal antibody which targets a key virulence factor in the bacterium Pseudomonas aeruginosa. And we're developing this as a long-term preventative treatment for patients suffering non-cystic fibrosis bronchiectasis, or NCFB, who are also chronically colonized with this bacterium. But there's also expansion options for this product into other indications, including chronic and acute respiratory diseases. Next slide, please, Pete. So starting with the patient's perspective and the impact NCFB has had on their life. So this is Scott's story taken from the British Lung Foundation. So like many NCFB sufferers, Scott was an athletic individual in his youth, taking part in numerous different sports. But when he turned 40, he started noticing symptoms of his lungs and started to develop recurrent lung infections. And he's now been taking antibiotics every day for nearly 20 years. He's now chronically colonized by Pseudomonas aeruginosa, and his lung capacity has really suffered and is now reduced to 55% of where it should be. During his more severe infections, he's found himself in hospital and he's had to have intravenous antibiotics. And over time, he's finding that these antibiotics have become less and less effective, and this is likely due to the emergence of antimicrobial resistance. Next slide, please. So what is NCFB and how does Pseudomonas aeruginosa relate to this lung condition? Well, NCFB is a chronic lung disease, and it's characterized by a chronic cough, the production of excess sputum, and patients often suffer from recurrent respiratory tract infections. And this is due to irreversible dilation of their airways. A frequent causative organism of these infections is Pseudomonas aeruginosa. And this organism is particularly problematic as not only is it intrinsically resistant to numerous antibiotics, but it also serves to activate the immune system in the lungs and causes a significant amount of inflammation. Collectively, this can cause the patient's exacerbations. So these are effectively a period of time where the patient's symptoms flare up. And these exacerbations can often require hospitalization and broad-spectrum antimicrobial chemotherapy or treatment with anti-pseudomonal antibiotics. And patients can experience many of these each year. So patients can experience, in severe cases, up to 1 exacerbation a month. And through recurrent use of broad-spectrum antimicrobials to control these exacerbations, we see that this serves to drive the emergence of antimicrobial resistance in Pseudomonas aeruginosa in the lung. So collectively, this results in progressive lung damage in the patient. And unfortunately for many NCFB patients, this also results in premature death as well. So the disease process in NCFB is frequently likened to a vicious cycle or a vicious vortex. So the nature of the lungs and the reliance on broad-spectrum antibiotics selects for Pseudomonas aeruginosa colonization and infection in the lungs. Once in the lungs, Pseudomonas has a variety of virulence factors, including the secretion of exotoxins, and these attack the immune system and prevent the immune system from clearing the bacteria from the lungs. The production of exotoxins in particular can degrade important immune cells called neutrophils and also leads to a significant amount of inflammation. And this collectively worsens lung function and causes further lung damage. So this worsening lung function only further predisposes the patients to recurrent infections, and so the cycle continues. So Respex serves to break this cycle. So not only does it provide better recognition by the immune system of the bacteria, leading to their clearance from the lungs, but it also physically blocks the virulence factor responsible for the secretion of these exotoxins into the lungs, and so prevents that destruction of neutrophils and inflammation that occurs as well. So next slide, please. So now I'll just hand over to Colm, who will tell us a bit more about the prevalence and impact of NCFB, as well as our clinical progress and development.
Thanks, Vicky. So this slide emphasizes the very large potential patient population where Respex could make a key difference to them. If you focus on the box on the top left, looking at US market, up to 300,000 plus patients with NCFB, again, even greater numbers in the wider European market, and even bigger numbers again in Asia Pacific, with up to 5 million patients diagnosed with non-cystic fibrosis bronchiectasis. And as you heard from Vicky earlier, a significant percentage of these patients get colonized with Pseudomonas. We know from international as well as UK data that up to 30% of patients get colonized with Pseudomonas, and the presence of Pseudomonas increases their risk of these flare-ups of their disease, or what we call exacerbations. And this has a real direct healthcare cost per patient. It's been estimated in the US that Pseudomonas colonization will have an incremental healthcare cost of over $30,000. And of course, the direct healthcare costs excludes the economic impact on patients and their families of time away from work, time in hospital, reduced quality of life. And so it's estimated that the current annual cost to the US healthcare system alone could exceed $3 billion from non-cystic fibrosis bronchiectasis. And that's excluding other chronic lung conditions that can also have Pseudomonas colonization, such as chronic obstructive pulmonary disease and asthma. And this isn't just some benign chronic condition, this non-cystic fibrosis bronchiectasis. There's up to 30% four-year mortality and 80% rates of hospitalization for patients with serious disease progression due to Pseudomonas. And then the box on the bottom left refers to the potential expansion into very large markets and other chronic lung conditions, such as chronic obstructive pulmonary disease, for example, in the US, with millions of patients, a clear percentage of these that have Pseudomonas colonization. Again, this is hard data, we know this. And also up to almost 700,000 asthma patients in the US may be colonized with Pseudomonas. Next slide, please. So this slide is about the clinical development, therefore, with the initial thrust of the clinical development being this repair non-cystic fibrosis bronchiectasis. We ultimately aim to reduce the frequency of exacerbations in these Pseudomonas colonized patients with non-cystic fibrosis bronchiectasis. We've made great progress in this. If you look at the blue panel on the left, we have actually completed our phase 1 studies. We've therefore shown that it's safe to give in healthy volunteers at 4 different dose levels. We know that the pharmacokinetics is very favorable with a long half-life of 30 days or more. And we know that we have had no significant adverse effects from infusion of this drug into these healthy volunteers at 4 different dose levels. And we're currently actively recruiting for this phase 2A study that was referred to earlier, where this is a first dosing of the drug in patients with non-cystic fibrosis bronchiectasis, with the key aim being to show that we know it's safe in healthy volunteers, so we are in the process of showing that it's safe in patients with non-cystic fibrosis bronchiectasis at 2 different dose levels. The study that we're currently recruiting for will also have bronchoscopy procedures for the patients where we take washings from their lung with a camera examination, and this will prove that the drug gets into the lungs. And of course, it gives us opportunity in these Pseudomonas colonized patients to look at secondary endpoints, such as the impact of Respex on Pseudomonas growth, sensitivity to antibiotics, expression of this virulent system that Respex blocks, and the efficacy of its ability to block these exotoxins that Vicky emphasized earlier are a big component of the damage to the lungs that Pseudomonas causes in these patients. And so once we finish this current phase 2A study, we then, we are already in fact quite advanced with our plans for the phase 2B study, which will be a clinical study in the UK with the primary outcome being attempting to show the reduction in exacerbations in a cohort of patients colonized with Pseudomonas who have non-cystic fibrosis bronchiectasis. And these patients will receive 4 doses of Respex at 3 monthly intervals over a 12-month period, with, as I said, the primary outcome being shown that we can reduce the exacerbation rate, which we know is a big contributor to the ongoing progression of lung damage. And we already have a number of major UK centres signed up to recruit for the phase 2B study. The protocol is very well advanced. We can't finalise that, of course, until the current phase 2A study is finished. And we have a series of secondary centres already expressed very keen interest to be part of this phase 2B study. And again, it's important to say that the current study that we're doing in Liverpool Clinical Research Facility is fully approved by the UK regulator and MHRA, and is in the recruitment phase, and we'll be ready for the submission of our phase 2B plans to the MHRA the second half of this year. And it's also important to say we have drug substance already manufactured for the phase 2 trials with 3 years stability data. So we're very excited about this program and the progress that we've made. Next slide.
Okay, so moving now on to our next program that we'd like to introduce to you today, which is MET-X. So this is a best-in-class broad-spectrum inhibitor of a really important class of antibiotic resistance mechanism called the metallo-beta-lactamases, or MBLs. And this agent works to restore the effectiveness or activity of beta-lactam type antibiotics, particularly in patients who are suffering severe infections caused by drug-resistant organisms. Next slide. So, of course, we know that antimicrobial resistance, or AMR, is a significant threat to human health globally. And thanks to the Lancet paper not too many years ago, we've now established that around 5 million deaths occur every year that are associated with AMR, and this is only predicted to increase. A particularly important new antibiotic resistance mechanisms are the metallo-beta-lactamases, or MBLs. So these were widely reported in 2009, and organisms producing these enzymes have now spread across the globe. And indeed, the World Health Organization has acknowledged MBL-producing bacteria as a critical priority, as there is currently no effective approved treatment for infections caused by these bacteria. So MBLs work to degrade or disable beta-lactam type antibiotics, which are vital antibiotics in our armamentarium, and also degrade really important antibiotics, our last line of defense drugs, the carbapenems. So these are often used for severe infections caused by multi-drug resistant organisms. So to tackle this key unmet medical need, Infex has developed a first-in-class broad-spectrum inhibitor of MBLs called METEX. And this works to effectively knock down or inhibit these enzymes, allowing the beta-lactams to effectively do their job and kill these MBL-producing pathogens. Next slide. So here we just have some proof of concept microbiology data. So we've combined METEX with meropenem in this study, and we've benchmarked this against a variety of marketed and developmental beta-lactam agents and beta-lactamase inhibitor combinations. And we've determined the MICs for 484 MBL-producing Enterobacterialis isolates. And the MIC90s have been determined. So this is the MIC at which 90% of the organisms would be inhibited. So really the take-home message from both this graph and the table on the right-hand side is that the MIC90 for all of the beta-lactam agents and combinations are significantly higher than the susceptibility breakpoint for either CLSI or UCAST. And the only exception to this, of course, is the combination of meropenem and METEX with a low MIC90 of 1. So this is within the breakpoint of meropenem and would be regarded as susceptible. Next slide, please. We've also explored the activity of METEX with a number of other beta-lactam agents in addition to meropenem, including cefiderocol, the combination of ceftazidime and avibactam, meropenem, vaborbactam, and also imipenem and relebactam. And using 27 MBL-producing Enterobacterialis isolates, we've determined whether these isolates are susceptible or resistant to these agents. So on the first graph here, we can see the number of treatment options that would be available for a given isolate out of these 5 beta-lactam agents. And for these MBL-producing pathogens, we can see that most of the isolates either have no treatment options or only one treatment option available to them. Next slide, please. So then when we combine these beta-lactam agents with METEX, we can see the situation is quite different. So all of these isolates now have at least one therapeutic option available to them, or for the majority, now 4 or 5 treatment options. And this is particularly important because not all of these beta-lactam agents are available in all countries across the world. So clinicians won't necessarily have this much choice in their hospital pharmacy. So it's important that METEX is developed with multiple different beta-lactams to give clinicians that choice, particularly for critically ill patients who are suffering from MBL-producing infections. Next slide. So now I'll just hand over again to Colm to tell you a little bit about our clinical development strategy for METEX.
Thanks, Vicky. So this is the clinical development strategy for METEX that I should emphasize has support from the European Medicine Agency via their Innovation Task Force mechanism, and METEX has also been granted qualifying infectious disease product status in the United States, and we have drug product already manufactured for phase 1. So the initial primary focus of our clinical development, you can see in the upper third of the boxes on the right, where it's METEX in combination with meropenem. Green boxes referring to the fact that we have already done an enormous amount of the preclinical work around the microbiology and toxicology and pharmacokinetic, pharmacodynamic data, and very close to being ready to go with early phase 1 trials showing safety in healthy volunteers, firstly of METEX on its own with single ascending dose and then multiple ascending dose studies, and then a further, of course, phase 1 healthy volunteer study combining METEX with meropenem in initially single ascending dose and then subsequently multiple ascending dose studies, and then a pivotal phase 2/phase 3 study in complicated UTI patients comparing meropenem with METEX combined with meropenem. And for this first indication of METEX combined with the existing antibiotic meropenem, we have a regional license deal in negotiation that Pete touched on earlier, where the licensee will pay for the clinical development and regional commercialization, with Infex receiving million pound milestone payments and double-figure royalties on regional sales. And of course, and because it's a regional deal that we're in negotiation, we retain the rest of the world development and commercialization rights, are retained by Infex for licensing. And of course, once we've progressed this initial indication for Metex combined with meropenem, we will be looking at Metex combined with other agents and work our way through both preclinical and early clinical development. Because as Vicky said earlier, this drug addresses a number of gaps in existing agents. So we're very excited about this clinical program, and we're very excited also that the European Medicine Agency have been particularly supportive of our development plans. Thank you. Next slide.
Okay, thanks, Colm. So finally moving on to our third program that we'd like to present to you today, which is COVAX. So this is our first-in-class oral pan-coronavirus antiviral therapeutic that is being developed for acute coronavirus disease as well as long COVID, and also in preparedness for future coronavirus pandemics. Next slide. So coronaviruses pose a significant risk to human health. So not only do they cause mild respiratory disease, they've also been associated with several significant pandemics throughout recent history. Of course, we'll all be acutely aware of the recent COVID-19 pandemic. So this very quickly spread across the globe in 2019, and to date has caused over 7 million deaths worldwide. Prior to that, in 2003, saw the emergence of SARS-CoV-1, or just SARS, if you like. This also led to over 8,000 infections occurring in more than 20 countries across the globe. And this carried with it a fatality rate of around 10%. Similarly, in around 2012, saw the emergence of Middle East Respiratory Syndrome coronavirus, or MERS-CoV. Again, this spread to over 20 countries worldwide, caused around 8,000 cases, but this carried with it a significantly higher fatality rate of around 35%. Next slide, please. So in response to this key unmet medical need, Infex has developed COVAX. So as I mentioned, this is a first-in-class oral antiviral drug, and this targets a key coronavirus enzyme called the papain-like protease, or PLpro for short. So this target is essential for viral replication, and it also has a role in evasion of the host immune response to infection. The target's conserved against the three key pandemic-related coronaviruses, MERS, SARS, and SARS-CoV-2, but it's also conserved in multiple animal coronaviruses as well. So not only is this agent being developed as a treatment for acute disease and long COVID, but it also has a place as a new line of defense against coronaviruses that have pandemic potential. And this element of the program is being supported by the US government agency, NIAID, or the National Institute of Allergy and Infectious Diseases. And as was mentioned earlier in the presentation, we currently have a first license agreement under negotiation for COVAX for use in long COVID applications. Next slide, please. So finally, some preclinical proof of concept data for COVAX. So the graph on the right-hand side shows a mouse model of SARS-CoV-2 infection. And what we've done here is we've taken two different doses at 100 mg per kg or 200 mg per kg and treated these mice and compared this to the marketed agent nirmatrelvir and compared this also to the no treatment control. And really what we can see is a significant reduction of viral load in the lung tissue, with the higher dose at 200 mg per kilogram achieving a reduction that is less than the limit of detection as possible in the assay for 5 out of 6 of the animals. We've also taken some samples from this model and subjected them to immunohistopathology, really to detect the lung damage that occurs as a consequence of SARS-CoV-2 infection. So if we take a look at the images on the bottom, in the saline vehicle image, we can see these brown regions in the image, and this is the lung damage that has occurred as a consequence of the virus. If we also take a look at the COVAX-treated samples, we can see no real visible lung damage present. And finally, we've also done some investigation of inflammatory markers, particularly those that have been demonstrated as clinically important for SARS-CoV-2 infection. And we see, looking at these inflammatory markers, interleukin-6 and CCL2, in response to COVAX treatment, we see an 80% and 94% reduction in these markers. Next slide, please. So that leads me to pass back to Pete to tell you a little more about the investment opportunity.
Thank you, Vicky, and thank you, Colm. Just a reminder, if you have any questions on the bits that Vicky and Colm have just covered, please pop them in the Q&A and we'll be able to try and field those after the end of the presentation. So moving just on to finish with more details of the company and the investment opportunity. So really, I mentioned right at the beginning, we're looking to secure 10 million pounds to cover the next 3 years. And that will deliver a number of milestones, both clinically and commercially for the company. So on Respex, you've heard already that we've delivered excellent phase 1 data. Phase 2A will be completed in financial year 25 and phase 2B in 26. And if all of that goes well, Shionogi's first license fee under the option agreement will be payable to us in financial year 27. On Metex, we've completed preclinical development package. The aim is to complete the first regional license agreement on that asset and start phase 1 in the next financial year and complete phase 1 global license, complete the phase 1 study and then move on to global licensing the year after that. And on COVAX, we've nominated the clinical development candidate plus backups and entered into the first NIAID contract to support preclinical development. We actually have a larger grant award under consideration from NIAID in at the moment. So our aim will be to hopefully secure that and contract for that grant funding in the next next financial year, and then complete the long COVID license and funding contract as has already been discussed. Then the year after, complete phase 1. Should note that just at the bottom there, our financial year is from August to July. So financial year 25 would end July 25. Then underneath all of that is the engine that generates our pipeline. So we have other clinical development candidates coming through. We have 3 applications in through the new UK PACE accelerator with 3 universities, and we're always looking to in-license, acquire, or develop new opportunities against very interesting targets. So short-term clinical and commercial milestones for investors who come in and share the journey with us on this round. Then, okay, so what are the milestones? So where will the 10 million get us to? So that will be completing Respex phase 2A and phase 2B, completion of the CTA enabling work and completion of phase 1 for Metex, complete the CTA enabling work and support phase 1 on COVAX, and developing the preclinical candidates. And that will get us to the significant Shionogi license option fee in financial year 27. So you'll see from the potential forecast income over the next 3 years, initially income from the Metex and COVAX deals, followed by a more substantial upfront payment from Shionogi in the year to July 27. So overall, that means we're seeking 10 million of new equity investment now to support the next 3 years and deliver these key program and commercial milestones. So just to summarize the opportunity at Infex before we go on to the Q&A, there's a significant market opportunity for the programs that we develop. We have a world-class capability with an outstanding team and a robust business model that's been tried and tested. We have partnerships already in place. The team's got an outstanding track record, supported by John Moulton and his investment. And we have significant support from other stakeholders, such as the grant awarding bodies, ICON, and a fantastic group of subcontractors and service providers, in particular at our home base in the northwest of England at Alderley Park. So with that, I would like to finish the presentation, and we can move on to the Q&A. So I'll stop sharing the presentation now.
Thank you very much, Pete. So, and thank you for everyone for submitting your questions. So the first one, I think maybe, Colm, if you want to take this, this is on RESPECS. Is the potential for RESPECS beyond NCFB significant in the future?
Yes, indeed. So we touched on this in some of the slides. So we know that a significant percentage of patients with chronic obstructive pulmonary disease are colonized with Pseudomonas. We know a percentage of asthmatic patients are colonized with Pseudomonas. We know a significant contributor to intensive care unit, hospitalized patient infections, hospital-acquired pneumonia, ventilator-associated pneumonia, that Pseudomonas plays a significant role in those patients, and also in bloodstream infections in critically ill patients. And while it's an overall small percentage of burns patients that get colonized with Pseudomonas, Pseudomonas is a big contributor to mortality in those patients. So there is really quite a big expansion. And we also think that cystic fibrosis patients, even with the advent of newer disease-modifying therapies, such as Kaftrio, that there is still a cohort of cystic fibrosis patients around the world who are Pseudomonas colonized, who are exacerbating with issues around resistance to antibiotics and toxicities from existing treatments. And so this is potentially game-changing for a whole bunch of disease areas beyond just non-cystic fibrosis bronchiectasis.
So, we've got another question, this time on COVAX. Nobody's talking about COVID-19 anymore, but surely the need for an antiviral against this virus is dramatically reduced. Is COVAX really needed now, or is it needed further in the future?
I guess I can start perhaps, and then Vicky or Pete can jump in. No, so okay, so I'm practicing respiratory physician, so I still am seeing people with significant impacts from COVID infections. Obviously, the particular risk groups are those who are unvaccinated or those who are immune suppressed. COVAX certainly is contributing much less to hospitalization, but certainly there are a percentage of long COVID patients where there's increasing evidence that virus, or viral RNA, viral proteins can persist, and there's, like I said, gathering evidence that persisting virus may be part contributing to a chunk of the long COVID. So I think there's, as Pete illustrated earlier, there's an enormous economic and societal impact from long COVID. So there is potential need for it sooner rather than later, but of course there's also an enormous need that we have medication sat waiting for the next pandemic, which most experts will say is not if, but when that will happen. And so, yes, I think there's a need both soon for COVAX, but also a longer term pandemic preparedness need. I don't know whether Vicky or Pete want to add to that.
Yeah, thanks, Colm. Yeah, what I think it's worth saying that the NIAID funding is under pandemic preparedness mechanisms, as would be the follow-on, and that there is strong interest in developing therapeutic options for SARS and MERS. And I think, Vicky, we also know that the PL-Pro target's conserved in a large number of animal and bat coronaviruses.
Yeah, that's right. Yeah.
Fantastic. Pete, this might be one for you. So you mentioned new AMR payment schemes. What is this and how does this work?
Yeah, okay. So one of the problems in the antibiotics world is AMR, which is the bugs becoming resistant to the antibiotics that we've known and used for decades. So we desperately need new drugs. But the problem is that as soon as you start to use them, we immediately start to get resistance. So the new drugs need to be conserved as much as possible. So drug stewardship is really important. And we only really need to use the right drug in the right patient to treat the right disease with the right antibiotic. So this means that the conventional way of selling selling drugs, so having a sales force and going out and trying to sell as many as possible, is just no longer allowed. So this is widely recognised. So we have these new de-linked systems that separate the reward from the amount of drug you actually sell, and you're actually rewarded to have a valuable drug that governments can effectively put on the shelf and use only when necessary. And I have to say that Colm played a major role in the development of the UK's thinking on that, so he could probably tell you a lot more about this than I can.
Yeah, so no, there's no question. Yeah, in fact, as Pete mentioned yesterday, the UK announced the sort of if you like version 2.0 or final if you like arrangements for dealing with new antimicrobials and those reimbursement mechanisms that will de-link volume of sales from reimbursement, which sends a signal that antimicrobial development is a valid area, even though these drugs often need to sit on the shelf waiting rather than have a large volume of sales. I was clinical lead for the initial project that informed this final system that the UK developed. And so there's been an enormous interest in that around the world, even though the UK are not going to fix that problem with the pipeline and reimbursement on their own. Clearly, the UK subscription model, where we delivered initial payments to two companies for two antibiotics, cefiderocol and ceftazimab-avibactam, with payments up to £10 million per year for up to 10 years, was an important signal. And this announcement yesterday is an even further signal and further encouragement for the country that we know have taken a lead from the UK, or the UK has given a lead to them about work in the US, Canada, Japan, Germany, Australia, various countries, and of course EU are trying to develop incentives at the moment that will drive more antimicrobial development.
Yeah, so I'm actually in Paris next week for a meeting between a whole lot of stakeholders in the EU and member states to try and move forward on the current proposals for EU-wide incentives for development of new antimicrobials. And I also help organizations to lobby in the US for the so-called Pasteur provisions, which are the US equivalent to what we're doing in the UK. And as I mentioned in the early part of the presentation, if you add up the potential contribution for Europe, US, UK, and other major markets, that will add up to a potential total reward of around $4 billion over the lifetime of the patents and market exclusivities.
Fantastic. This might be another one for you, Pete. What is the investment process, and when are you looking to close the round?
Okay. Yeah, so we actually launched formally the investment process and started emailing people with introductory decks around 2 weeks ago. There are a couple of investors that we've been talking to for a bit longer than that to potentially provide a cornerstone for the round. But we opened a data room up yesterday, so we have a data room available, and if anyone would like to arrange a follow-up to this, an initial one-to-one meeting, please get in touch, and then under NDA we can let people get into the data room. The round as a whole is being supported by John Moulton's team at MGL And Adrian Field there is also talking to individual investors to bring a investor syndicate together. And of course, any existing shareholders will be available, will be welcome and able to support the round as well.
Got a couple of questions here on Respex. I don't know, Vicky, if you want to take these. Can Respex be used with traditional antibiotics?
Yeah, great question. So how we envisage using Respex and how it's being developed for use is really as a maintenance therapy for NCFB. So this will be given, so Colm mentioned the long half-life, so we envisage it will be given infrequently to patients, and that will provide them with protection against Pseudomonas and help them better clear Pseudomonas from their lungs. But yes, it can be used alongside traditional antibiotics. So we would still foresee that the existing standard of care, those anti-pseudomonal antibiotics that are given during an exacerbation, would still be used. And in fact, we have some really promising preclinical data that shows that Respex can work synergistically with antibiotics. So in preclinical models of infection, we see that the mice are better able to clear, and we see better survival rates using the combination of Respex with those anti-pseudomonal antibiotics as well. So potential synergies there.
And another question on the phase 2b study for Respex. Is the study statistically powered to detect a reduction in exacerbations with 38 ARM? And if so, what percentage reduction would you expect to see versus placebo?
Yeah, I'm guessing that means 38 patients in each arm. Yeah, so that's 80% powered. We've run the calculations ourselves with a preferred CRO and also with colleagues from the clinical trial unit in Liverpool. Of course, we're constantly developing and looking at that in the light of the experience that we have in the phase 2A, and depending on the level of biomarkers and the rate of exacerbations that we see in patients. That will allow us to see a 25% signal, so 25% reduction in number of exacerbations in the 12 months. But we'd be hoping to see a larger signal with that than that. Colm, I don't know if you want to add any more to that.
That's a really good summary. And as Pete said, the powering was based also on published data of known exacerbation rates from trials that have been published. And we know these Pseudomonas colonized patients, we know from the European data and worldwide data that most of the Pseudomonas colonized patients will have 3 or more exacerbations a year. But we've been more conservative than that because exacerbation rates have been a bit lower in some of the published clinical trials. We've tested the appropriate signal of, Pete mentioned the 25% reduction with key opinion leaders in UK, Ireland, mainland Europe, Japan, and the United States. And that's a valid, felt to be a valid reduction in exacerbation rate. So, we haven't just pulled these numbers out of the sky, that they are based on quite a lot of input from published data and key opinion leaders. But as Pete said, we will, obviously as the phase 2A data comes through around impact on biomarkers and whether the PK could be different in patients as opposed to healthy volunteers, then there could be adjustments off that phase 2B trial. So we can't finalize the plans for that just yet.
A question here on Metex. Would you need to generate preclinical and clinical data for full spectrum beta-lactamase before filing for approval, or would you lead with meropenem and submit the data for additional beta-lactams as label extensions as the data becomes available?
Yeah, that's a really good one. The plan is to generate data initially, as Colm indicated on the slide deck, from Metex plus meropenem, but we've had really good initial conversations with the European Medicines Agency, and I think there's a lot more that we can say on this, but that'd have to be done under an NDA, because obviously those conversations and those plans are confidential at this stage. Colm, I don't know if you want to say any more than that, though, as little as we can say, really.
I really don't. My initial comment was going to be that yeah, we have to be careful what we say because some of it is commercial in content.
So please
we're very supportive of plans going beyond Metex meropenem. I suppose that we can say that. Yeah.
Yeah. So please get in touch, and we can talk about that in more detail.
And one final question on Respex. Has Respex been used in any human tests with people with fibrosis?
Maybe I should jump in there. So apologies if this wasn't clear from the earlier slides. So basically, we completed our phase 1 study of dosing of Respex in healthy volunteers. We're currently recruiting for that first study to give Respex for the first time to patients with non-cystic fibrosis bronchiectasis. But obviously, the term fibrosis relates to many different types of lung disease, but so put simply, the mechanism of the drug is against Pseudomonas specifically rather than an anti-fibrotic medication. So the simple answer to that is no. It's a drug targeting the organism to reduce its ability to cause damage and toxicity to the lungs and reducing its ability to persist and restoring the ability of the immune system to control and potentially eradicate the organism.
And again, to be clear, the drug's only been in healthy volunteers to date, and the phase 2A that we're initiating at the moment, recruiting for at the moment, will be the first time in Pseudomonas colonized NCFB patients.
Fantastic. And that's all the questions. Pete, I don't know if you wanted to say a few final words.
I would thank you all for attending. Thanks, Louis, for hosting this today, and Colm and Vicky for taking us through the presentations. Please do get in touch if you'd like to follow up. We can arrange one-on-one conversations. Vicky and the team will be more than happy to go through the more detailed technical presentations on any of the programs. We do have a data room in place if you'd like to explore further investment in the company. And so I'd just like to thank you for your attendance today and wish you a very good rest of the day.
Okay, thanks everybody. Thank you.
Thanks all.
Thanks everyone.